Hi, my name is Will. For my biology role model project, I chose somebody who I think most scientists would admire. She had a passion, she devoted herself to it, and as a result, she has been involved with some absolutely groundbreaking discoveries. This is Dr. Elizabeth Blackburn. She was born on November 26, 1948, in Tasmania, Australia. Here, she's pictured with her 6 siblings. She's in the top left corner, second from left. Although she has kept her personal life pretty, private, as not much is known about her hobbies or interests, her research and schooling is very well documented. She developed an interest in science from a very young age. Her initial interest in biology was rooted in animals, something that I can personally identify with, and it was this curiosity about animals that led her to pursue a career in biology. She took a pretty traditional path, graduating from the University of Melbourne in 1970 with a bachelor's in biochemistry, followed by a PhD in molecular biology from the University of Cambridge in 1975, along the way being faced by and overcoming the stigma surrounding women in STEM. It was at Cambridge where she began her research in DNA, which led her to a postdoctoral project at Yale about DNA sequences. It was in this research where she had made her largest discovery, specifically her discoveries of telomeres and telomerase, while researching chromosome and chromosome replication. As she worked to further her research, her focus has been on answering the questions of how telomeres and telomerase work functionally and how they influence cellular aging and other diseases, which is research she has now been working on for almost the entirety of her career. And in 2009, she actually won a Nobel Prize for her work. And this is what I find most admirable about Dr. Blackburn. She's devoted herself so fully to her work, and in what I think some would say is a relatively niche area of research, but because of that, she's allowed us to make such great advancements in our understanding of both medicine and biology. I admire her passion. Her most recent publication focuses on shorter telomere length and antidepressant responses, the importance of which lies in gaining a better understanding of how telomere length influences psychiatric disorders and mental illness. Using outpatients from a University of California hospital, researchers used blood samples to test telomere length and telomerase activity over the first 8 weeks of a subject using antidepressants. The figure you see now is from that publication. For context, and I'll try to keep it pretty simple, telomeres are regions of DNA at the end of chromosomes to prevent the chromosome from deteriorating. Telomerase is an enzyme responsible for maintaining telomeres. Telomeres naturally shorten as they wear down during DNA replication, and telomerase rebuilds them, which, by the way, are facts that we only know thanks to Dr. Blackburn's research. This figure shows telomere length, TL, and telomerase activity, TA, in terms of their specificity and subject sensitivity to antidepressants. It shows a relationship between short telomere length and low telomerase activity, with low responses to antidepressant treatments. This is similar to a lot of the work she's done, experimenting and recording functions of telomeres and the implications of those functions. As of now, she continues her work to better understand telomeres at a molecular level, and the long-term goals of this research are to discover more of the secrets DNA holds, and possibly develop treatments for diseases such as Alzheimer's or cancers, treating mental illnesses such as depression, and some hope that understanding telomeres will even unlock the secrets to aging. Included here is a quote from Dr. Blackburn. "If you fail, is it really a failure?" These are my references. And these are my image reference hyperlinks.